Hello, biology. The purpose of this video is to give you an opportunity to see some of the types of questions that you're going to see on tomorrow's final. Again, my apologies for not being able to host a Zoom and allow students to participate in this as I'm making the video. But I had forgotten that I am helping to supervise the graduation this afternoon, and will not be available right after school today. So again, because this is a video, you can pause, fast forward as often as you would like. For the 100-point questions, I'm going to give you about 30-45 seconds. For the 200-point questions, about a minute and a half. For the 300-point questions, about 2.5 minutes. And for the 400-point questions, about 3.5 minutes. Give you an opportunity to find this information on your study guide, try to come up with the answer, use your book to look up some of this stuff. In any event, I hope that you find this useful. And of course, good luck on tomorrow's final. All right, here we go. Let me get my timer set up. All right, here we go. We're going to do one column at a time. So we're going to do all the cell division stuff. This is from meiosis, which was in chapter 11, and mitosis, which was 10-2. Random stuff's all over the place, of course. Protein synthesis, that was 12.3 and 12.4, if I'm not mistaken, from the book. Genetics was 11.1, 11.2, 11.3, I believe. And of course, evolution was chapter 15, 16, and 17. All right, ladies and gentlemen, here we go. Mitosis produces two identical daughter cells, while meiosis produces blank, genetically different daughter cells. Should be an easy one. It is a number that I am looking for. About 5 more seconds. All right, here we go. So mitosis, two identical daughter cells. Meiosis, because there are two divisions, doubles that number of daughter cells, so you get 4. And remember, they're genetically different daughter cells because you have the opportunity for crossing over to take place in prophase 1. Let me get the timer ready. Okay, here we go. If an organism's diploid chromosome number is 46, its haploid chromosome number is what? So you're going to have to know what diploid and haploid mean. Mitosis produces blank daughter cells, while meiosis produces blank daughter cells. These are not numbers. Which produces diploid? Which produces haploid? All right, a couple more minutes for this one. About 1 minute. $30. About 10 seconds. 5, 4, 3, 2, 1. All right, here we go. Okay, so remember, diploid is double, haploid's half, so the haploid number would be 23 chromosomes. Of course, mitosis produces diploid daughter cells. That makes sense because body cells need all of the instructions to do whatever that body cell needs to do. Haploid comes from meiosis, and that also makes sense because you're going to get half of the chromosome number in the sperm cells from the father. You're going to get the other half of the chromosome number in the egg cell from the mother. Together, they're going to make 46 chromosomes, and that's why the the baby, the human, will have characteristics of both parents because they get half of the chromosomes from the dad, half of the genes from the dad, half of the chromosomes from the mom, half of the genes from the mom. All right, here we go. If a body cell has 20 chromosomes, how many will it have after mitosis? Didn't we just talk about this? During cell division, what phase will the chromosomes become visible within? The cell's volume, the cell's surface area, and the rate at which food, water, and other nutrients move in and out are all limits to what? Which phase is a period of cell growth between cell divisions? About 3 minutes. About 2 minutes. About 1 minute. Okay, about 10 seconds. All right, here we go. Okay, so if we're talking mitosis, you start with 20, you end with 20. Prophase is the first phase of mitosis and meiosis, and that's where the chromatin condenses into noticeable chromosomes. Cell volume, surface area, and the rate at which it brings things in and out all control or all are limits to cell growth. And then that period of cell growth in between meiotic or mitotic divisions is called interphase. That's where the chromosomes are doubled during DNA replication, and it's where the cell prepares for mitosis or meiosis. All right, switching gears here. So we're going to do all of these columns first. We'll do the random stuff last. All right, let me get this back here. Okay, during blank, RNA molecules are formed by copying DNA inside the cell's nucleus. This is a process. Processes in science end in TION or SIS typically. All right. Here we go. Transcription. Transcribe means to copy down. So transcription happens in the nucleus, and then messenger RNA leaves the nucleus through a nuclear pore. It goes out to the ribosome in the cytoplasm, and that's where translation or protein synthesis, protein building actually takes place. Okay, here we go. If messenger RNA's codon is UCG, what will transfer RNA's anti-codon be? See if you can remember the rules of base pairing. Five seconds. All right, here we go. So U and A go together, C and G go together, and G and C go together. Remember, wherever we're talking about messenger RNA or transfer RNA, there will be no thymine. U will always replace thymine. All right, here we go. The process where modern organisms have descended from ancient organisms and have changed over time is called what? Phosphate groups, guanine, cytosine, and adenine are all found in both DNA and RNA. True or false? So this is obviously random. About 45 seconds. Ten seconds. All right, here we go. Okay, so modern organisms coming from a common ancestor, changing throughout time. is evolution. And true, phosphate groups, guanine, cytosine, and adenine are found in DNA and RNA. Remember, uracil is only found in RNA. Thymine is only found in DNA. Ribose is only found in RNA. And deoxyribose is only found in DNA. Genes contain instructions for making what? A group of individuals that can interbreed and produce fertile offspring are called what? First one is protein synthesis. Second one is a random question that comes from chapter 16. About 20 seconds. Ten seconds. All right, here we go. Genes are instructions for proteins. Genes are read, transcribed, translated during the process of protein synthesis. And a species is a group of individuals that can interbreed and produce fertile offspring. All right, here we go. On to genetics. When Mendel realized that the short plants return in the F2 generation, so we're talking about the pure breeding cross, he thought that the allele for shortness and the allele for tallness blanked during gamete formation. About 10 more seconds. Oh, here we go. He believed that they segregated or they separated, means the same thing. So remember, this is the classic cross where you had two purebred parents, one that was tall, big T, one that was short, little t. The F1 generation was all big T, little t, and of course that is tall because tall is the dominant allele or the dominant gene. But then when he self-pollinated that F1 generation, and he had big t times big T little t in the F2 generation, short individuals came back. Only 1/4 of them were short, but the only way for that to happen is if the big T got separated or segregated from the little t when sperm and egg cells were created. And that's the process of segregation or the principle of segregation. All right, here we go. The likelihood that a particular event will occur is called blank. When Mendel crossed white and black chickens, the offspring were speckled. This showed that the white and black traits were what? About 40 seconds. 10 seconds. All right, here we go. So that's called probability, the likelihood that an event's going to occur. And oh my goodness, this is incorrect. Hold on, this is co-dominance. My bad. Co means together. That means both of the dominant traits are going to be together in the offspring. Incomplete dominance is when you have a blend. It's red flowers, white flowers, you get pink flowers. So, my bad on that mistake. Co dominance. The principles of blank can be used to predict the traits of the offspring produced by genetic crosses. If you cross two hybrids, big T, little t, what fraction of the offspring will be short? We just talked about this. About 30 seconds. Here we go. So the principles of probability can be used to predict traits of the offspring from crosses. All right, so 1/4 of the offspring were short, 3/4 were tall. The short ones were little t, then half of them were big T, or heterozygotes, and then 1/4 were big T. Of course, the phenotype of being tall is the same for big T, and big T, little t. All right, here we go. Which one of Mendel's ideas describes the fact that alleles for seed shape segregate without influencing alleles for seed color and do not influence each other's inheritance? Whose ideas include all of the following? Genes are passed from parents to offspring. In most sexually reproducing organisms, each adult has 2 copies of each gene. Some forms of genes or alleles are dominant, and some are recessive. About 1 minute. About 30 seconds. Ten seconds. All right, here we go. So independent assortment basically means that it's kind of like when we were doing the 2 trait cross problems. You had big R, big Y, little y. You have to account for all the different combinations, big R, big Y, big R, little y, little r, big Y, little y, because when the chromosomes get line up for metaphase, they line up randomly, which means you could have any possible combination of genes or alleles. So that's independent assortment, and of course, those were all things that Mendel came up with. All right, on to evolution. The fact that Darwin thought fitness is the result of adaptations, the ability of an organism to survive and reproduce in its specific environment, and that certain characteristics give organisms a better chance of survival all support the process of blank through blank blank. The following are all benefits of what process? Offspring never resemble parents exactly. There's an increased chance of evolution due to natural selection. There is an increased probability that good combinations of genes will be produced. About 1 minute. Five seconds. All right, here we go. Okay, so those are all things that are happening through. Well, let's complete the sentence. Better chance of survival all support the process of evolution through the mechanism or the tool, natural selection. And sexually reproducing organisms never resemble the parents exactly. There's an increased chance of evolution through natural selection because you get an increased chance in variation. And there's also increased probability that good combinations of genes will be produced that would lead to survival, reproduction, passing on those genes to your offspring, which over time would be evolution. Whose experiments showed that organic molecules could have formed from inorganic gases and lightning before life was present? Adaptive radiation, punctuated equilibrium, mass extinctions, coevolution, convergent evolution are all important topics of what? A little over a minute. Five seconds. All right, here we go. Okay, so that was Miller and Urey's experiment, and those are all topics of macro evolution or evolution on a very, very large scale. Okay, what type of isolation is necessary for the formation of a new species? According to Darwin's theory of blank, the individuals that tend to survive are those that have variations best suited to their environment. True or false? If an organism dies without reproducing, its genes leave the gene pool, and the frequency of alleles for those characteristics decrease. Okay, about 15 seconds. All right, here we go. Okay, so reproductive isolation has to take place before speciation can take place. And remember, there are a lot of kinds of reproductive isolation, a lot of types of isolation that lead to reproductive. Behavioral isolation, temporal isolation, geographic isolation, all can lead to reproductive isolation. Darwin's theory of natural selection indicates that individuals tend to survive and have the best variations suited to their environment. And then, of course, over many, 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 many generations of natural selection acting on these good characteristics and acting on the bad ones by not allowing them to survive, it leads to evolution. And true, makes sense that if an organism that doesn't have good characteristics dies without reproducing, the genes for that bad characteristic, or maybe just a characteristic that isn't so great, leaves the gene pool, and that decreases the frequency of those characteristics. So over time, you'll have fewer and fewer, and eventually none of those characteristics, and it'll be replaced by characteristics that are good for the environment those organisms are living in. All right, here we go to the random stuff. Different forms of a gene are called blanks. A blank blank can be used to determine the probable outcomes of a genetic cross. About 30 seconds. 10 seconds. All right, here we go. Different forms of a gene are referred to as alleles, and there are dominant alleles and recessive alleles. And Punnett squares can be used to determine the probable outcomes of a genetic cross. All right. If an organism is big T, little t, this is called what? If an organism is tall, this is called what? Fitness is a result of species what? About 2 minutes for this one. About 1 minute. About 20 seconds. All right, here we go. Okay, big T, little t is a genotype. Now, of course, big T, little t would give you a tall phenotype. And fitness is a result of a species' adaptations or characteristics. Remember, fitness is the ability to survive and reproduce. So if you've got good adaptations, good characteristics, it's going to give you a better chance to survive and reproduce. Whose genetics ideas apply to all sexually reproducing organisms? Who explored the Galapagos Islands, observing variations in the characteristics of the organisms living there? Which type of dating would be used to find the absolute age of a fossil? All right, you guys, a little over 2 minutes. I'm going to step away. I need to use the restroom real quick. All right, I am back. About 30 seconds. Okay, 5 seconds. All right, here we go. So Mendel's ideas of genetics can be applied to all sexually reproducing organisms. Darwin was the guy on the Galapagos, obviously. And radioactive dating is the more specific type of dating. Remember, relative dating is just, oh, this fossil's older than this one, but younger than this one. Radioactive dating can actually determine the exact, well, within 1,000 years or so, the exact age of the fossil, which of course tells you when those organisms were actually living. All right, here we go. Some more random questions. The two main sources of genetic variation are what? Mendel's principle of blank states that some alleles are dominant and others are recessive. About 1 minute left. .30 seconds. Okay, here we go. Two main sources of genetic variation are crossing over, which is the same as genetic shuffling. This happens, if it happens, during prophase 1 of meiosis. Mutations take place during DNA replication. And then it's the principle of dominance states that some alleles are dominant and others are recessive. All right, organisms with two identical alleles for a certain trait are called what? When Mendel crossed red flowers with white flowers and got only pink offspring, this showed what? And all of the combined genetic information in a population of organisms is called what? 30 seconds. Ten seconds. All right, here you go. All right, two identical ones are homozygous. If they have a big T and a little t, they're heterozygous. Incomplete dominance. So what this means is that the red trait is dominant, the white trait is dominant, but you don't get either one. So that means that they're incomplete dominance. Incomplete in between. Pink is in between red and white. All the combined genetic information in a population of organisms is its gene pool. Last questions, kids. What organic molecules are made by constructing long chains of amino acids? Mutations are always blanks to genetic material being copied. Which phase has the chromosomes gathering at opposite ends of the cell while losing their distinct shape and a nuclear envelope or a nucleus membrane starts to reform? 'Bout a minute. Thirty seconds. Okay, 5 seconds. All right, here we go. So proteins come from the monomers, amino acids. Changes to genetic material, any change to genetic material is classified as a mutation. Some mutations are good, some mutations are bad. Mutations over a long period of time, though, do give the potential for evolution. And during telophase, the last of the four phases of mitosis and meiosis is when you have the chromosomes that are at opposite ends of the cell. You have the DNA is starting to be surrounded by a nucleus or a nuclear membrane. And of course, if we're talking about mitosis, cell division is about to be over. If we're talking about meiosis 1, those two daughter cells are going to go ahead and divide another time to form four daughter cells. Hi, ladies and gentlemen. I hope you found that useful. Study really, really hard. This will give you an idea of the types of information that you will have on the test, not the exact questions. And remember, these questions are harder because you don't actually see the answer. The test should be significantly easier because you do see the answer. You just got to know which one it is. All right, ladies and gentlemen, we will see you later.